Hello and welcome. In this tutor, we will see different organelles. Here is our tubules with attached ribosomes called rough endoplasmic reticulum, and without attached ribosome are called smooth endoplasmic reticulum. Then we will talk about interplay between endoplasmic reticulum and Golgi apparatus. Proteins are particular type of secretion are produced by endoplasmic reticulum, and then they are transferred as a transport or transfer vehicle to the Golgi apparatus, where they are packaged inside the cells and sorted to the different locations. Like they can be exocytosed from the cells and they can be used inside the cells, and they are also help production of the lysosomes. Like we see the lysosome productions in the same way. Rough endoplasmic reticulum produce enzyme of lysosomes. Then they are going to the Golgi apparatus, where they are packed inside the physical. Now lysosome is ready to play its actions. Then they are the food molecule is endocytosed inside the cells, and lysosome is attracted to it and release its particle of hydrolytic enzyme inside it. That will cause the breakdown of food particle into the simpler ones, and this is how phagocytosis will take place. Now let's us talk about other organelles like microbodies. These are the diverse category of organelles containing different type of hydrolytic enzymes and different type of other enzymes that are not present in lysosomes. These are called microbodies. The example can be included in animals. They are peroxisomes. Peroxisomes are membrane-bound organelles containing hydroxylases, catalases, peroxidases enzyme. Peroxidases called formation and degradation of hydrogen peroxides. Hydrogen peroxide is produced. It can cause oxidative stress to the cells, and it is also cause aging of the cells, and it is cancer producing, and it has very harmful cells. And to overtake this, peroxisome also has catalase organelle enzyme. Catalase enzyme break down the hydrogen peroxide into the water and oxygens, which are both beneficial to cells. Then there is the mitochondria, powerhouse of cells. We will study about briefly, not more detail. In more detail we will study later. It is the double membrane organelles. You can see it is outer membrane. Then there is the inner membrane having many folding inside the cells, and this is called as Christie. And there are many type of the enzyme on it for the production of the ATP, like electron transport chain that help produce ATP. And Christie also increase the surface area for the function of the particular type of uh, reaction of the enzyme. And inside there is the matrix. Matrix contain its own DNA, own ribosome, circular DNA, and its ribosome is 70s like that of bacteria and prokaryotes. And because they produce energy, they are called powerhouse of cell. Let's talk about cytoskeleton. The cytoskeleton, as human also needs skeleton cell do so. They consist of microtubule, microfilament, and intermediate filaments. And most of the microtubule, intermediate filament, and microfilament give the flexible and protective framework to cells and hold the structure of the cells in place. Here is our mitochondria. Let's talk first about it. Here is outer membrane, here is inner membrane having manifolding nucleus, and inside is the matrix of mitochondria. Here is our cytoskeleton, given proper framework to the cell and giving its proper size, and it contain microtubules. These are the radicular microtubules. Then there are intermediate filament, and these one are the intermediate filament attached to the inner side and outer side of the plasma membrane. Then there are the microfilament, and they are more cylindrical in cylinder in shape. Means their diameter is very low. Then microtubules, they are hollow and cylinder and cylindrical structures, but they are not as cylinder as that of the inter microfilament or intermediate filament. They are in having greater in diameter, but they are cylindrical in the shape in animalism, and they are spiraling subunit of globular protein like tubulin subunits. The protein present in microtubule is tubulin. What is the function of the microtubules? They help in movement of the organelles inside the cell membrane. Organelles are moving and moving inside the cell because of microtubules. Then there is the movement of secretory vesicle. It is like exocytosis and endocytosis. Secretory vesicle are moving from endoplasmic reticulum to Golgi apparatus, and from Golgi apparatus to outside of cell. All this is possible because of the secretory vesicle, and secretory vesicle are moved because of the microtubules. Then there is the movement of chromosome during the cell division. It is also takes place with the help of microtubules, and they are also part of the transport system within the cells, like nerve conductions, and then they help movement of materials inside and outside the cells. Then so they are important part of cytoskeleton, and they maintain the shape of cells, and they can change during the period of specialization and help in proper shaping of the cells. Then there are intermediates. Their cylindricity or diameter is intermediate between those of microtubule and microfilament. There is no particular type of the protein, but they are composed of heterogeneous group of the proteins, and they are different in different type of cells. What are the functions? They help maintain a cell shape, and their function is also maintenance of cell shapes and special organization of the organelles. They also help promote the mechanical activity within the cytoplasm, and compartmentalization may also take place. And microfilament, they are composed of also called actin filament. They are most found in muscle cells. They act together with the myosin to form muscle contractions, and they may also be formed in non-muscle cells. And in non-muscle cells, they provide mechanical support for the various cellular structures and help in contractile system responsible for cell movement, like amoeboid movement is because of microfilament, and some protozoal movements. Then we will study about cilia and flagella, which help in movement.